I'd like to welcome everyone to the ISV's monthly webinar series. This is the second installment of our series for this year. Today we have Dr. Marcus Buggert from the Karolinska Institute, is going to talk to us about his current research. Before we get to that, I'd like to make a few announcements. First of all, the webinar series is now being listed on the ISV website. If you'd like to see future topics and speakers, please check out the website. In addition, I want to let everyone know that the Congress for the ISV will be taking place in October 21 to 23 in Seoul, South Korea. Registration for that Congress will open up in April, so please check the website for updates. And then lastly, I just want to let you know this recording, as well as others, will be listed on the ISV website for those who did not make the meeting today or in a time zone that's inconvenient. You can see this webinar presentation again online. So I have the opportunity here to introduce Dr. Buggert. He is from the Karolinska Institute. He joined in 2018. He is currently an assistant professor at the Karolinska Institute, where he focuses on memory T cells associated with infectious disease and cancer. So we're lucky to have him present today. His paper was selected as our paper of the month. And so it's nice that he has the opportunity to talk about that as well as other research activities. So welcome, and I turn it over to you. Please feel free to share your screen and begin.
All right, perfect. Can everyone see this? All right, that's great. All right, perfect. So first of all, thank you very much. Thank you, Ted, very much for the invitation to give this talk. So as the title says, well, what I'm going to do is to talk about T-cell immunity to SARS-CoV-2 after mRNA vaccination. And it's going to be divided into two different parts. So the first part, I'm just going to go through some of the studies that we and other people have done to look into correlates of control after breakthrough infections, as well as what happens in immunocompromised individuals after they get vaccinated with mRNA vaccines. But then I'm also going to focus mostly on the second half of the paper of the month, as you just called it, from December from last year that we published in Science Immunology. So this is, very generic background. I think that everyone knows this already, but just to get a recap for everyone that is not aware of what a T cell or a B cell is, they are the two different parts of the immune system, the adaptive immune system. They form what we call the memory of the immune system. And antibodies are part of our humoral immune responses. I'm just going to laser pointer here. And basically what they do is that they bind to the virus so they can't bind into the receptors, and it neutralizes the virus before infection takes place. Whereas T cells, which is also called the cell-mediated immune response, they instead recognize antigens or presented on MSC molecules, MSC class 1 or 2 molecules, depending on if it's a CD4 or CD8 positive T cell. And what these cells can do, particularly in the case of CD8 positive T cells, is that they can launch a cytotoxic attack. They release these molecules that eradicate virus-infected cells. That is, key correlate for protection also in cancer, but they're also, of course, involved in autoimmune conditions as well. And what is unique, I would say, about T cells, and particularly when it comes to mRNA vaccines, that these vaccines are slightly different in comparison to what other vaccines, for example, inactivated vaccines have been in the past. And the reason for why I'm saying that is because these vaccines are very good to generate both B and T cell responses, and particularly what we call CD8 responses. So this is old data from John Verry's group, essentially showing that people that got vaccinated with mRNA vaccines versus those who had a prior COVID before who got vaccinated. And these are data that has been generated by a lot of different labs, that throughout the course of vaccination, you can see that you generate good antibody responses. So slightly higher, at least in the beginning, these individuals which has what we call hybrid immunity. I'm going to come back to that a little bit later when we talk about this other paper. For the second part, memory B cell response is also very well generated, but as you can see, memory T cells are generated to a very high degree as well. And many of these studies, what they've been doing, including us, has been characterizing these responses to understand what they are, particularly when it comes to T cells. So we know that after the early phases of a priming and then boost strategy here, you can kind of see by, again, following naive versus those that are SARS-CoV-2 recovered individuals, that with time, it seems to be that individuals that have only been vaccinated, they will actually come up to the same levels magnitude-wise, both when it comes to CD4 and CD8 T-cell responses. But what is unique, and this is actually something I'm going to come back to and show a little bit later on as well, is the fact that what seems to be happening is that you generate quite good CD4 responses early on, which is important also for generating a coordinated human immune responses. But with a boost and the more booster shots you seems to be getting, you're actually augmenting your CD8 responses as well. So I'm going to come back a little bit to this later when I'm going to show some data across different immunocompromised states also to see that this is a feature that is not only taking place in healthy donors. But in general, when we think about vaccines, to come back to this point about the correlates and what we usually think about vaccines, I think there's no question about that. We all know here that a robust neutralizing antibody response is highly associated with protection after vaccination and a natural infection. However, as you can see here on the right side, we all know that there's reduced neutralization against Omicron and the different variants of Omicron, which is causing less protection from getting the actual infection. However, the vaccines are still very effective, particularly against severe disease in healthy donors. So why is that the case then? There's probably a lot of different explanations for it, and one explanation can be that we have still good non-neutralizing antibodies that could provide some kind of protection. But there is quite a lot of data, and I'm going to highlight some of them actually soon as well, which is showing that T cells, what they can do very efficiently is to cross-recognize the Omicron variant. This was actually the first paper that was published on this, which came from our group. It took us about 1 month to put this together from the time point when we knew, from the time point when we conducted our first experiments until the paper was published 1 month later. But basically what it showed in a very simplified fashion is that if you stimulate T cells with wild-type peptides from the original ancestor strain, the Wuhan strain, versus stimulating the same cells, essentially at least, with the Omicron strain, you see very little differences in vaccinated individuals, in convalescent individuals, and these data has then been confirmed by a lot of other papers, including some of them listed here. But in addition to that, probably hundreds of other papers showing that T cells are efficiently cross-recognizing multiple strains. This data was from the initial strain that came from BA1, that came from Omicron, but we have recently also shown in a paper that was just published in Cell Host and Microbe now that T cells efficiently cross-recognize also hypermutated variants such as BA286. This is data from 2 different cohorts, essentially where we did comparisons of the Wuhan strain response, the BA286, and then BA1. And essentially what we can see is that in vaccinated, non-infected, those with hybrid immunity, which are infected in 2 different cohorts, we can see the same kind of patterns. So essentially what it means is that for new hypermutated variants, we most likely are still going to have efficient cross-recognition by T cells unless there is a substantial shift happening in mutation patterns. So do we think that these cells are important? I was kind of stressing that before that I was going to come into that, and I'm going to just highlight a couple of different papers, this one a little bit more in detail. And the reason for why I'm doing that is just to represent for you the fact that when we start to look into different, or when not we, but when other people have been looking into responses very early after breakthrough infections, correlating antibodies, B cells, T cells, different type of T cells, what you can see is that a very rapid T cell, and particularly CD8 positive T cell response, is closely linked to early control of SARS-CoV-2. And this is studies that have something that you can see very early on, that the viral decay rate basically is correlating very narrowly to the anamnestic, so the highly activated T cell response afterwards demonstrated in the blood. This is not the only sort of correlate studies that has been done of early SARS-CoV-2 control. There's been a couple of other studies, including this one from John Verri's group. This one is a challenge study in Science Immunology. basically showing very similar features, that particularly the increased activation of CD8 plus T cells seems to be a very good correlate of early control of SARS-CoV-2 infection at breakthrough. But there's also other studies showing that even that these responses can provide protection from exactly what we were saying before, from cross-recognition, like a recent paper in Nature Immunology, and similarly after mRNA vaccination, and particularly it seems to be associated with that if you have airway surveillance of T cells, that seems to be associated with the fact that you have better clearance. There's also correlation in immunodeficient patients, 2 papers in Science Translational Medicine, excellent paper, both of them. I think both of them came out last year, essentially showing that in B-cell deficient individuals, if you have T-cell responses, you're going to reduce the risk of severe COVID-19, and those individuals that has a very strong T-cell deficiency has a hard time to defend themselves and clear the viral infection. So I thought now, like I said before, is that what in the beginning of the pandemic, everyone was interested, of course, to look into mRNA vaccines and how they induce particularly antibody responses, but also when it came to T cells, people were looking into healthy donors. But what we shortly thereafter became interested were to look into those individuals that we thought was going to be at the increased risk of getting infection. And as I just told you, with those previous papers now coming out, it tends to be so that if you can induce a good T cell response, because in some individuals, as you will see, you don't have any B cells because of different underlying diseases or because of treatments, it seems to still be so that those T cells can kick in. But what we were really interested about was to initiate a cohort where we were studying the immunological response following mRNA vaccination in immunocompromised individuals. So we did this. This was an open label phase 4 multicenter study that we initiated early 2021. We included healthy controls, primary immunodeficiency disorders of different varieties, HIV infected patients, hematopoietic stem cell transplanted, even CAR T treated individuals, SOT, so solid organ transplanted people, and also those with chronic leukemias. And essentially, the initial paper was a safety and efficacy trial where we were showing that 72% of the immunocompromised patients were seroconverting. This was similar to what other people saw because we also could see that seroconversion rates were found at lower degrees in solid organ transplants and in chronic lymphocytic leukemia. And what was quite interesting is that when we started to look into T cell responses in the same patients, actually what we find is very similar patterns. Individuals from all groups can induce detectable CD4 positive T cell responses. You can see in some of these individuals, like these ones here labeled SOT, so that's solid organ transplants, they indeed, after 6 months, had a much lower response in comparison to healthy donors, but also many of the other groups as well. Particularly those ones in CLL were the ones that also were lagging behind, just like the antibody data was showing before. However, by looking at this data a little bit more in detail, what we could find is the fact that there is a good correlation between T cell responses, specifically now on this axis is CD4 responses and antibody titers. There is a good correlation here, but you can also see that there are some people which are in this quadrant up here, which are individuals with T cell responses, without antibody responses. And what is interesting here that we were able to do was to kind of probe back into some of these individuals, because some of them were entirely lacking even mature B cells, like these 3 patients here with X-linked agammaglobulinemia, XLA patients, which we were able to follow them through a little more in detail. I'm not going to go into this in detail, but this is just to demonstrate what we can do, I think, if we think this through in a very powerful way by doing human experimental immunology. And that is essentially to vaccinate individuals to study the immune system. So this was something that we could do here through this cohort because these individuals had never seen this antigen before. So we're looking into healthy donors versus XLA patients. We did then a single cell RNA sequencing, and without going through too much into detail, because this is a very complex pattern, which the idea is not to show you everything in detail, but what you can do with these type of technologies is to really now assess in greater detail whether they have a Th1, Th2, Th17 response, TFH, whatever you want, essentially. But essentially, the conclusion of this data was the fact that even without mature B cells, actually these individuals were able to induce very potent T cell responses. So this is essentially like a human knockout model to study that in greater detail. And if anything, we could actually show that these individuals in general were able to generate as robust and maybe even to some degree more robust T cell responses than other individuals. So we next kind of asked, how do T cell responses then evolve over time following mRNA vaccinations in immunocompromised patients? So in order to study that, we continued to follow these individuals also during the Omicron wave. These individuals got a first and second vaccine shot, then a third and fourth one, where we then assessed them during this 12-month period. And we didn't look into the same amount of patients as we did in the initial trial, partly because of dropouts, but also because of availability of cells. But I think one thing you can probably see by looking into this plot here is this fact that, so this is A masses here. We have CD4 responses on the top, CD8 responses here. You're looking into CD69 versus CD4 ligand here with a negative control, CD69 versus 4-1BB here. And what you can kind of appreciate is that from day 0 to 12 months, these responses are really going up over time. And this is true both for CD4, but particularly for CD8 responses. And by looking through all these different cohorts here, essentially what we can find is that indeed, over time, you can find for PIDs, HIV, HSCT, SOT, CLL, and healthy donors, you can really see that both against the wild type, the Wuhan strain, but also against the Omicron strain, we can find increased responses over time. Just to simplify this a little bit, because it's a very complex plot to look at, you can focus in on PID, so that's primary immunodeficient individuals, and HIV infected donors. You can kind of see, just like I showed in the slides before, that even immunocompromised individuals, you can actually indeed see that the responses between the Wuhan strain versus the Omicron strain is quite similar. It's if anything, it's like I was saying before, I think it's like 9% reduction with sometimes some p-values which are significant, but that's because all the bars are kind of going down the same direction. But one thing which was exciting and quite interesting for us to see was kind of coming back to one of these other points that I was saying before in healthy donors, was this fact that some individuals which were kind of lagging behind a little bit, and particularly when it came to CD8 responses, like this SOT group here, actually, when they got booster shots, many of them now had detectable CD8 responses. So we can kind of see that by looking at the median CD4 net response that it increases quite a bit here, but you can see the same thing in particular for CD8 responses. And just to put this into some kind of perspective, because one might ask, what does all this mean basically? You have stronger responses. But let's say now they have 1% of your T cells in the body, which are now recognized as spike. And you have approximately 400 billion T cells circulating in the adult human body. If now 1% of them are suddenly spike specific, it means that 4 billion T cells in the human body now can recognize the spike antigen. And I think that is really the difference, of course, from what we are now versus where we were before, when we talk about that people have better protection from getting severe disease versus what they did when COVID hit us 4 years ago. One thing which is kind of highlighting what I said before as well, but it's depicted in a little bit different type of plots here, is that when we look into those individuals that were responding better now to the boosters, we saw that those individuals that responded quite poorly in the beginning, such as SOTs, they are the ones that are gaining the most from booster shots. So basically, if one focus on this plot here, if one looks at the 12-month versus 6-month net response versus a correlation versus 6-month net response, that means that those individuals now that had a pretty poor response at 6 months, they are the ones that actually gained the most from getting the boosters. So with those shots, they actually are, with time, they are getting better T cell responses. And it's not only something that we see with T cells, actually the antibody data is very similar in this regard as well, particularly when it comes to a certain group, such as solid organ transplants. And this data here on the right side is kind of just depicting that, again, in a little bit different shape by saying that those individuals that had a poor response at 6 months, those individuals had similar responses at 12 months versus those other ones. And those were the ones that got 4 doses. Does anything of this mean anything? Like I said before, there is some other studies which has now been doing correlative studies to it as well. We're able to do something similar as well, because what happened during this 12-month phase were the fact that the infection with Omicron was hitting Sweden as well. And you can kind of see that during this phase here, we were getting a lot of these samples. So many of these individuals that are quite severely, one would say, immunocompromised potentially, were actually now, after all, generating pretty good antibody and particularly T-cell responses afterwards. For instance, these individuals with B-cell malignancies. And of all these individuals with a disease score, which is 1 to 10, 10 means mortality, individuals, there was none of them even that was ICU treated. A few of them, 2 of them or 3 of them, were essentially taken into hospitals and needed oxygen, but none of them succumbed due to COVID during this time point. So, and the majority of patients experience only mild symptoms. Again, emphasizing this point that even if they became affected, it seems to be the fact that actually there's something with their immune response, whether that is T cells, antibodies, whatever. It seems to be something that is at least keeping them in check, at least during this period of time when we followed them. All right, so like I said before, but that was the part about immunocompromised individuals. So what I thought I'm going to do now is to shift focus now to this paper that we talked about before, which was the that came out in December and became the monthly paper of December, I think, 2023 for ISV. It's this paper that we published in Science Immunology on SARS-CoV-2 vaccination, and particularly mRNA vaccination, and that it enhances effective qualities of spike-specific T cells that are induced now by COVID-19. So this is essentially a story which is centered on this terminology about hybrid immunity, which I mentioned a little bit before. And for those that doesn't know what hybrid immunity is, this is immunity through both vaccination and infection. And this is depicted here in a paper by Shane Crotty, who essentially showed that if you look into memory B cells, antibodies, CD4 and CD8 T cell responses, that depicts a tree here. You can see that the natural immunity and vaccine immunity, they have a shorter tree in comparison to this tall tree, which you see after hybrid immunity. I think this, this difference between them has to some degree been erased now when people have gotten more booster shots. But in the beginning of the times, one could really nicely show, particularly when it came to antibody responses here, that natural infection plus a single dose even of a vaccine were generating very potent and robust antibody responses. And one might say, is this important or not? I think it is. This is one paper out of many, I would say, but this is one paper which essentially is compiling data from different sources. So this is a systematic review and meta-regression paper, which looked at the protective effectiveness of primary SARS-CoV-2 infection and hybrid immunity against Omicron infection and severe disease. So essentially, if one focus now on the blue lines, which is hybrid immunity, that means that people have become infected and then they got a primary series of or one booster shot versus naive individuals. You can kind of see that when you look into any infection, it kind of decreases over time as a consequence of that new strains have developed, Omicron strains, which are evading neutralizing antibodies, and these are decaying also over time. But if you look at hospitalization or severe disease, you can see the hybrid immunity is really, on a higher level still afterwards. Again, whether that is a consequence of antibodies, T cells, or a combination of them, maybe that we have better mucosal responses, I think is a hot topic of debate to some degree at least. But what we thought was interesting based on this data that was emerging was the fact that when we were vaccinating our individuals at the same time point, people started to become concerned. They said, okay, but what will happen if we boost our individuals or if we boost ourselves over time? what will essentially be the impact on the quantity and quality of T-cell immunity following repeated vaccinations against SARS-CoV-2 after you already had an infection. So we'll repeat the SARS-CoV-2 vaccination post-infection fuel, for example, T-cell exhaustion, which is a very hot field in chronic viral infections, and particularly in cancer disease. So we kind of had that answer in the back of our heads. And we were also interested about it from this context of looking into individuals who had previously had mild versus severe disease. Because in the beginning of the times, there were individuals that thought that people who got severe disease of some reason, maybe they got an exhausted immune system that would lead them to be prone to get the infection in the future as well. So we thought that, okay, let's look into individuals that became infected during the first wave of infections. So this is about 4 years from now, mild and severe individuals. And then we look at them 6 to 9 months afterwards in a convalescent phase. Oops. And then afterwards, we look at them post mRNA vaccination again at 18 months. And we conduct a lot of different type of assays to answer these questions. So this is again AIM assays, just demonstrating the similarities and differences when it comes to DMSO controls versus spike. This is not something surprising. Essentially what you see if you look into CD4 responses, mild 6 to 9 months versus 18 months here is that it's increasing. The same is seen for severe individuals as well. And this is true for sure over time when you look at this. And you can actually probably also see that severe individuals, they have stronger responses in comparison to what mild individuals have. And this is already there implicating that actually individuals with severe disease, they have T cells that can respond to the vaccine. They're not exhausted or allergic or something like that. They're actually able to expand very efficiently. And just like we would expect us to see, what we find is that if you look into nucleic capsid, membrane envelope, or 3 to 10, you can see a reduction of the CD4 responses over time because these are obviously not part of the vaccine. So that was CD4s. What about CD8s? Actually follows very similar trajectories as well. This is again AIM assays we're conducting here, looking at CD69 on the y-axis, 41BB on the x-axis. You can kind of see the same patterns here over time, that both when you look at mild versus severe individuals at convalescent phase, they have differences, but actually the differences, if anything, is even higher afterwards, again implicating that individuals that had previously severe infection did not have allergic or exhausted T cells. And indeed, when you look into spike-specific T cells at 6 to 9 months, so in other words, after convalescent versus after vaccination, you do see that this is increasing over time. So far, so good. But what about this question that we were asking in the beginning about what happens when you get these booster shots? Could it be so that the quality of the response is going to change? And we had already started to get some implication of it by looking into spike versus non-spike responses in other cohorts, that there could be some differences when we start to look into some inhibitory receptors. And indeed, and in order to do these assays, what you need to do was to use tetramers, which is not stimulating the cells and upregulates many of these inhibitory receptors. And indeed, what was a little bit puzzling for us when we started to look into SARS-CoV-2 spike-specific tetramers here, and we looked into markers such as PD-1 and TIM3, indeed, actually, when you look at convalescent versus after vaccination, you can actually see an increase of PD-1 expression. You don't find it for non-spike. You don't find it for CMV-specific responses. The same tendencies are true for TIM3 here as well for spike-specific responses. And even though it's a little bit maybe less clear, I believe, than PD-1, but that kind of made us a little bit concerned because it could mean that with time, when you get exposed multiple times to the vaccine, it might exhaust your T cells somehow in the end. But it turned actually not out to be true. And actually, if anything, it seems to be the opposite. Because what we found as a paradox to the PD-1 part of the story is actually that after vaccination, it seems to be that antigen-specific T cells, spike-specific T cells specifically, they have increased functionality for CD4 plus T cells in this case, in terms of interferon gamma, IL-2, TNF, the triple production of these in mild individuals. And the same thing is true for severe donors. Again, implicating this point that even in severe individuals, there's no T cell exhaustion, although IL-2 is maybe not going up as much, even if there is clear tendencies of it. But really what this is implicating here for CD4 plus T cells is that what we see for PD-1 expression is a little bit different. Looking at CD8 plus T cells, which was what we were doing before with tetramers, the tendencies is the same. So if you look into functional virus-specific T cells against spike here, by looking into these interferon gamma positive cells here, we actually do see that if you look into triple productivity of cytokines, you can indeed see that, for example, TNF, granzyme B, and interferon gamma positivity, they are of the interferon gamma positive cells, they are indeed increasing after vaccination, both in mild and severe individuals. So we thought, okay, this is interesting. So let us, and these cells also retain functionality after up to 5 booster shots, because we can confirm that by looking into individuals, particularly immunocompromised individuals as well. But then we thought, okay, let's do the same thing to what we did before. And that is to use an unbiased platform and use single cell analysis of spike-specific T cells now to look into the same features with this platform instead of flow cytometry. So what we did were to take convalescent patients before and after vaccination. We stimulate the cells with peptides. We oligoconjugate these with site-seq molecules. We sort them based on AIM markers, and then we conduct single-cell RNA and TCR sequencing. And essentially what you find here after deconvoluting the data is that you have spike-specific CD4 and CD8 positive T cells here in different clusters. And indeed, by including site-seq molecules that measures your protein expression at the single-cell level, we were kind of able to confirm the other data. And this is now showing that PD-1 expression particularly for CD8 positive T cells it's increasing post-vaccination. But what is interesting, similar to the other data that we were showing before, is that when you look into CD4 responses, indeed, by looking at all these different clusters of CD4s, but you actually see it for every single cluster as well, is the fact that there are certain molecules changing, and the top one changing is actually interferon gamma. So it seems to be that the mRNA vaccines are potentially maybe reprogramming the T cells to become more Th1-like, for instance. We don't know that for sure, but the data implicating at least that at this point. But for CD8 plus T cells, it was actually even a more concrete shift, one could say, because here it tends to be so that the cells before and after vaccination, they're almost entirely shifting from this cluster 4 here until cluster 2, both in mild and severe individuals over time. And these clusters are different in terms of interferon gamma expression being much higher on this cluster 4 here, and granzyme B expression, just like the flow data was showing before. And I mean, again, it sort of means that independent on inhibitor receptor expression, it seems to be that these vaccines are somehow modulating these cells to actually become more responsive and gain increased expression levels of certain effector molecules such as interferon gamma and granzyme B. Finally, is this something very puzzling? Is this something that is new? I guess in humans, to some degree, it is, I would argue for, but it's there is some data in mice which is very interesting. This paper came out for about a year ago from Dave Massipus' lab. For those who doesn't know what this is one of the frontiers within resident memory T cell development, and he had this very interesting way of doing transfer experiments where they did 51 successive immunizations over a 10-year time point into mice. And essentially one of the features of what they found with time is not so that these cells are developing functional exhaustion. They retain their functional capacities, but it also is indeed associated with increased levels of PD-1, TOX, and some of these other molecules which are associated with exhaustion. But probably what this implicates is that there's a difference of chronic antigen stimulation versus getting repetitive stimulation, but with some resting in between. And that is essentially what they're showing in this paper, and it kind of aligns with mRNA vaccines maybe as well, or vaccines, I would say in general, is that if you get some resting time between the time points when the T cells are seeing their antigen, it might actually lead to increased expression of some inhibitor receptors, but not functional exhaustion. And I think that is the difference also to take with us to some degree when we are studying these responses for the future. If people then become concerned when they see increased PD-1 expression and some of the other inhibitor receptors, that unless you have looked into the functional outputs of these cells, it's not what I would call functional exhaustion. All right. So the summary is that this is something I've been trying to say during the entire pandemic, that for me, immunity is not, a black or white phenomenon. Protection from disease happens through different layers of adaptive. It can happen through innate immune layers as well, but these different layers of antibodies that are protecting us from getting the infection, but you can also have protection from severe disease, which is associated, I would say at least, to a fairly high degree with T cell responses. And the reason for that is because preserved protection from severe disease after vaccination infection is partly mediated via cross recognition by memory T cells, because they can recognize all these new variants as well. We see that there is improved T cell and antibody responses in immunocompromised patients over time. That is also associated with the fact that these individuals seems to be a little bit less prone, at least, to develop severe disease. And finally, we show that vaccination after infection in this kind of hybrid immune setting, it increases the magnitude, indeed, both in mild and previous severe disease, but it also increases the quality of the T cell responses despite this kind of increased PD-1 expression over time after mRNA vaccination, which I think is more, as I was talking to in the end, is due to the fact these cells are getting re-stimulated, but they're still functional because they get this resting state in between, which is different from getting chronic antigen stimuli during functional exhaustion. So that is it essentially from me. I have a lot of people from my group to thank for this work, particularly Curtis Kai, who was the first author on this last paper, together with you, Gao. Thomas Müller, who was first author on the other papers in STM on immunocompromised individuals, people from the Karolinska University Hospital, from University of Pennsylvania, Loyola, Cardiff, our funders here in Sweden and internationally, as well as patients and healthy volunteers. Thank you very much.
Thank you, Marcus. That was fantastic. If people would like to ask questions, please raise your hand and I can unmute you and let you ask your question of the speaker.
You want me to stop sharing?
You can if you like. It's up to you.
Yeah.
I thought I saw a hand raised, but it went away. All right.
There's one
There's a question in the chat. You want me to read it, or you can read it?
Could you please comment on the impact of pre-existing T cell responses to endemic coronaviruses and SARS-CoV-2 T cell immunity? Yes, it's a great question. It's something that we, I guess, and a lot of other people have been working on as well. There's data we have a paper, for example, in kids showing that individuals who have been more recently exposed to different human coronaviruses, which are endemic, seems to have a better functional response against SARS-CoV-2 as well. If that is if that is something that could help them in the end, maybe that's the case, but we don't know that for sure. There are some data on this, obviously, papers from Malamaini and others showing that you can have these polymerase specific T cells potentially that could maybe abort infection as well. I think it's tricky. It's hard to know for sure if that is a correlate or not. There's actually linkages as well. It seems to be some HL linkage there to certain B molecules, B15, I think it is, and B7 as well, where there are immunodominant epitopes from endemic coronaviruses that potentially could play a role. But it's, usually I say like this, that it doesn't at this point of the pandemic, it's less important, I would say, probably, because now when people are vaccinated, of course, it's more important that people have that type of immunity rather than having protection from endemic coronaviruses. But I think it's an interesting field. I think it's super interesting as well to study this also, particularly in different age groups. But I don't know for sure to give the short answer to this question if it's so important anymore as it was maybe when people were naive to SARS-CoV-2 in the beginning.
Okay, you have another question in the chat from Betty Kapman.
Yeah, children tend to have milder disease than adults. Exactly. So we have been doing some of these studies. It seems to be that the kids are, it's a little bit tricky, these studies, because kids have we have only conducted these studies when we have been doing studies on pre-existing responses, and there you can clearly see that children, they do have a more functional, sometimes even more robust uh, cross-reactive SARS-CoV-2 response. After these kids, however, get the infection, it seems to be that they have a lower response in comparison to adults. But it's tricky because the reasoning for why kids probably have a lower response than adults is because they had less viral loads, and that triggered fewer T cells, and people have seen the same thing with antibodies as well. But it seems to be so that there's a functional component here that is slightly different between them that potentially could play a role. But we haven't looked so much into that, kids versus elderly, I would say, or kids versus adults. We've been looking a little bit into adults, and there's and elderly people, and there seems to be some functional differences as well, but not so much into children, more than the pre-existing immunity paper I was talking to about before.
All right, your next question is, given your results, do you think it would be relevant to include other proteins in the vaccine, or is T cell responses against spikes sufficient?
I mean, we've had these discussions with different companies even, and I think that, I mean, personally, I would think that that would be a way forward. And I know that BioNTech even has a, clinical trial because they had a Cell paper showing that you get, improved protection from severe disease if you include nucleocapsid, for example, responses in your vaccine design as well. I think, however, that without knowing too much about it, that the companies feels at this point that the vaccines are working quite well already against severe disease. So for that reason, I don't know if Pfizer and BioNTech are thinking about approaching this to the market yet, but they may. But and of course, I mean, I personally, I would think that that would be great, and particularly people with B cell deficiencies. But we'll see if that happens or not in the future.
All right, a follow-up question. Is there evidence that mRNA persists in some vaccines, and does this impact the T cell response versus other vaccines?
Yeah, I mean, I don't think that there is plenty of evidence suggesting that mRNA is persisting in a human body for that long time. I mean, that's the point about mRNA, is that it gets, degraded quite fast. But however, it seems to be that given the fact that there's a quite high antigen load that is being generated, it seems to be that people have ongoing germinal center responses for quite some time afterwards in draining lymph nodes. And that is something that Ali Alibadi and others with him have been showing. We don't know for sure if that is causing anything when it comes to T cells, but I would say like this: if it would be persistent antigens taking place after mRNA vaccination, then I would argue for that we will see more of what I was talking to in the end of functional exhaustion, but we don't tend to see it. And if anything, actually, we tend to see the opposite, that these responses becomes more functional and more robust with time. So I don't think that the mRNA is persisting, but but maybe some proteins that are generated by the mRNA are present in draining lymph nodes afterwards. Yes.
All right, so the data has shown that hybrid is better than just infection. Do you believe that ultimately, after several rounds of infection, the same set points are reached?
Yeah, maybe. It's hard to say, right? I mean, I think that they're kind of offering different good features, if one can say. I mean, my other life, I usually talk a lot about tissue residency, And that is something that I think most people really believe that infection is actually inducing. It could, of course, be so that with multiple rounds of infection that you will actually then, build up your immunity and you will build it up against different proteins, and particularly in the respiratory tract and in the circulation, that it will become as good immunity as the vaccine. But what the vaccines and the mRNA vaccines are doing very good is that they are boosting the antibody responses very efficiently, and it seems to be that they're maybe modulating the T cell response as well. I think it's a fair question. It's something that we haven't studied. It would actually be interesting to study. We have talked about it, but yeah. I'm not saying I'm exhausted to do to COVID, studies. I think that this could be interesting as a follow-up study in the future as well, but we haven't planned it at this point. But it's a valid question. Yeah.
Very good. So do you think that inflammatory T cell responses are associated with enhancement of any disease?
I think that it depends on what one means with inflammatory T cell responses. If we talk about them in terms of primary, okay, so primary COVID, when people have not been exposed to the virus before, there is data implicating that T cells are playing an active role because they get in there too late, basically, and then they are part of the rest of the immune system to cause collateral damage, essentially, of the entire immune system. Then the question is whether it can enhance other diseases. Maybe. I mean, there's a big field now trying to study, long COVID even, and to understand if T cells can be part of that after vaccination. But we don't know for sure, I would say, at this point. No.
So do you have an opinion on what's the best time to wait between booster shots?
Oh, I mean, usually before I was saying that one should wait at least 3 to 6 months. I mean, now it's hard to say. I would probably say at least 6 months, but maybe 1 year as well. It depends on how healthy one is, and what one wants to achieve essentially with the booster shots as well. If one wants to achieve, of course, getting full protection from getting infected, well, then of course it's good to get the newest vaccine all the time, with, of course, the caveat there being that we're always one step behind the virus. So it seems to be that we are never able really to generate, enough neutralizing antibodies to get that protection. But I would say that for a normal person, that's, I don't know, once per year, but one can potentially even wait longer. But if
Right.
one really wants to boost the system, yeah, of course, every 6 months, maybe even, if you really want to get these high titers of antibodies, yeah, why not?
So there's a question about your immunocompromised studies that you performed. Were there any autoimmune participants on therapy other than B cell depletion therapies such as TNF inhibitors? And do these
No,
therapies have any effect on the T cell responses to vaccination?
yeah, so it's a good question. There's a couple of studies which has been done on TNF alpha inhibitors, which doesn't seem to have too much of an impact on a T cell response, which is quite interesting actually, because it also seems to be that yeah, that individuals which are on TNF alpha inhibitors doesn't seem to be more prone to get severe disease. But there are some studies on that with Bertoletti and others, but we did not look into TNF alpha inhibitors. We were looking into, so solid organ transplant patients, they're on what is called MMF. So that's a drug which are basically they are blocking the proliferation of both B and T cells. So that's the reason why they have a hard time to make good antibodies. But most of the other people, they were either primary immunodeficient individuals which had B cell deficiencies, or people who were CLL patients who had B cell deficiencies, and some of the other ones also. So I would say it's a mixture of B and T cells, but not any TNF alpha inhibitors. No.
So have you seen any evidence of antigenic drift in the spike CD8 T cell responses?
Um, well, it depends on what's meant with it, but I think that I mean, to some degree, we do see that a new infection with, for example, Omicron, it can actually create new targets of it, which is quite interesting, but it's not substantial, I would say. I think that the response, however, is more created against other viral epitopes, against other antigens, rather than a true antigenic drift in the spike T cell epitopes. So I would say, yeah, maybe partly, but not to, you know, full degree of me feeling comfortable to say that we really do see that, just like you would expect it to see it with some flu antigens, for example, for antibodies.
So you may not have had this opportunity in Sweden to look at this kind of study, but there's a lot of different types of COVID vaccines out there now in the world. Have you looked at any heterologous vaccination strategies between an mRNA and a different type of vaccine, and does it seem to make better T cell responses?
Yeah, this is a good question. Some studies have been conducted, particularly in the UK, where they have done that because they were vaccinating them with adenovirus and then with mRNA vaccines. And I would say about 25% of our individuals in the beginning were vaccinated with adenovirus vaccines, but afterwards, and afterwards with the mRNA vaccines, we have not looked into that because we choose to follow mRNA vaccinated individuals from the beginning. And after that, they have been continued given that. The only thing I can say there is that some people have received Pfizer and then Moderna afterwards, but it's not true heterological challenges. But it seems to be true that at least when it comes to antibodies, yes, you may be able to boost it better. We don't know for sure when it comes to adeno and mRNA vaccines if that can proficiently boost a good CD8 response. I think there is some studies on it, but they came out for some time ago now, and I don't remember exactly what the results were. But we haven't looked into it in detail, no.
So how does hybrid immunity affect the imprinting of the immune response and shapes different T helper responses post-vaccination compared to people that weren't previously infected before they got vaccinated?
Shaping the bond citizen reactions different to vaccinations. I mean, so what seems to be clear is that when you get infected, you of course are going to have a Th1 response that you generate. And we have been quite curious about this also in our data set, but we have not really been able to answer it. If you, with the mRNA vaccines, when we see this shift going on from into this Th1 profile or more cytotoxic CD8s, whether it's new clones that we are generating or whether we are now boosting the clones which are already present there. And I think the data out there is kind of suggesting that it could happen, that both could happen. And actually, we are kind of seeing the same thing in our data set as well. But it seems, but I would more argue probably for that the response is modulated. So I don't think it's a hard imprinting of the Th1 response or Th17 response or whatever one want to call it after infection. So I don't feel comfortable to say that the infection is causing an imprint. If anything, I feel that it seems to be polarized towards one subset, could potentially be something that we haven't measured specifically. Maybe these cells are producing more IL-10 after infection, and then what happens after mRNA vaccination is that we're kind of shifting that more towards the Th1 response in the circulation and probably TF8 response when it comes to the lymph nodes. But we haven't seen anything that points towards that there's an imprinting of a specific population after infection that can't be changed by the vaccination. But it's a good question.
So one of the questions here is, do you think the exhausted T cell is a reversible state, or is it permanent after repeated vaccination?
I mean, no, so, and that's the point. This is what I want to emphasize, is that these cells are not exhausted. I mean, it's different. So these cells, even if they express PD-1, they're still functional. I mean, it's, and the reason for that is because they get rest in between them. I know it's complex. We always talk in our group if we should give these cells a terminology or something like that, but which looks exhausted, but they're functional. There's a lot of T cells in our blood which are PD-1 positive. They're not exhausted because of that. Exhaustion is true. When that happens, it's an epigenetic different kind of route that cells take very early after their developmental task. But it also happens because you get this chronic infection, chronic antigen stimuli going on at that early time point. So we do not see any evidence for that. And I think that John Verry and other people are, confirming that, even if I know that some group have been claiming after infection that you may get an exhausted profile of your cells. I don't feel that our data is supporting that. The opposite, if anything, it seems to be that these cells are able to expand both after mRNA vaccination, but also after infection very well, to be honest, even if they have slightly higher levels of PD-1 on them after some time. But I think that's more a phenotype because they have received some stimulation more than one time point rather than that's functional exhaustion. But it will be very interesting to look more into that epigenetically, just like Dave Masterpuss and other people have done in that paper that I showed before.
All right, we're reaching the top of the hour, so I'm going to ask this final question here is, do you have any data on whether long COVID patients have similar or altered T cell profiles compared to those that don't have long COVID?
Yeah, so there's a lot of data on this. I mean, this person can probably wait for, we talk about a week or so, and we will publish a paper, not publish a paper, we have a preprint on this. I mean, the only thing we can say is that we don't see, at least, a big reduction in terms of the magnitude of the responses. There is, however, for non-spike specific T cells, and I think it's important to say that it's non-spike and not spike, because non-spike is now going to mark the infection rather than people maybe getting infection. There it seems to be that if you look very carefully, there's tendencies maybe that they have in long COVID slightly higher levels of some of these inhibitor receptors and a combination of them that could imply that they have met more virus at some point, maybe some persistent virus as well. But it's tendencies that are significant, but it's not clear cut, I would say, like a chronic infection like HIV or something like that, where you really see a difference versus what you would see in this scenario between long COVID versus non-long COVID. But we see many other interesting features in this preprint that will come out soon.
We look forward to that.
Yes.
Okay, one last quick question. So in your
Yeah.
immunocompromised participants, patients, do you face any challenges with low numbers of T cells since they're immunocompromised? Does it affect your assays and how you interpret your results?
I think, well, it's a fair question. I guess because what we're doing right now is that we're looking into percentages as well. And indeed, if we would look into absolute counts, it might be some differences to what a healthy donor might have. And I think to some degree, that's the reason why I've been a little bit careful by saying it's fully restored immune response in comparison to other individuals. But one difference there is that what immunocompromised individuals have received is multiple booster shots in comparison to our healthy donors. And I think when they get that, with time, it will not immediately, but with time, they're going to get up there to those levels, which are fairly similar to healthy donors. But surely, I mean, we don't know for sure if these responses are as good and as high magnitude versus what a normal healthy donor would have, and particularly if you start to look into tissues and stuff like that. No.
All right. Well, thank you very much, Dr. Buggert. We appreciate you giving this presentation. It was very fascinating. There was a lot of comments, a lot of discussion. And so
Yeah, great. Yeah,
to,
never received this much comments, but great. I mean, honestly, like it was great. It was fun to answer all these
we look
questions.
forward to your upcoming publications. We thank everyone who stayed online. We, in the March presentation, will be about Dr. Trevor Smith from Inovio. And so we'll see you in a month. Thank you. Take care.
Perfect. Thank you. Bye.